I work as a biologist full-time and as a conversation enthusiast part-time. I just love to engage in conversation so much that I was just standing backstage and, instead of fixing my mic or something, I was trying to overhear what was going on stage. So, super guilty. So when I had to choose my master's research, an opportunity came up, which was to explore some hidden conversations, and they were part of a murder mystery, as you can figure out by the photo there. Now, it's top secret murder investigation, by the way, so I will spill the beans, but it's meant only for the 500 of us, okay? Not a single person more. Great. Now, I know we've all had an eventful but yet long day, right? So why don't all of us just sit back, relax, and take a deep breath in and out. Perfect. Feeling better? Yeah? Really? Because I just had all of you inhale thousands of microorganisms. Exactly. So each time we breathe in, we actually inhale thousands of microorganisms. And the recent COVID-19 pandemic has taught us one and only one thing, which is to stay away from microorganisms as far as we can, right? So the fact that there are microbial cells inside our body sounds like a big reason to worry, right? But don't worry, because your immune system is at a constant battle with these microorganisms to ensure that you remain disease-free. Now, however, unfortunately, in certain conditions, your immune system can get weakened down, thus giving these bacteria an upper hand at winning this battle and causing disease. Now, here's where my thesis comes in, because I dealt with a similar situation where at one end of the battleground stand patients with a lung condition known as bronchiectasis. This is the third most prevalent inflammatory lung condition, and I quote, is one of the most neglected respiratory disorders in the world in research. Imagine that. And because it's inflammatory, it means that the immune system is always active, which is not ideal either, right? I mean, instead of fighting just pathogens, it starts fighting the own lungs of these patients and breaking down the lung proteins into amino acids. And if we were to zoom into the lungs of these patients, we would see that their airways are full of thick and sticky mucus, which neither of us want, do we? So now on the other end of the battlefield stands a bacterium, which unlike the neglected respiratory disorder, is basically like the Shah Rukh Khan of the microbial world. I mean, you can see the resemblance, right? So this bacterium has been given the title of highly critical pathogen by World Health Organization. And from eyes to lungs to hot tubs, it's everywhere. So you know where I'm getting at, right? But why am I so interested in bringing these two entities, which are at the completely different scale of fame, together and diving into their conversation? What's the why of my research? Here's where the murder mystery begins. The reason why bronchiectasis patients die, the number one reason why these patients die, is not because of bronchiectasis. It's because of infections by this bad guy over here. The bacterium loves the lungs of these patients so much that it just lives there chronically despite rigorous antibiotic therapy. So we wanted to understand what are these strategies this bacterium, this bad guy over here, is using to persist in these lungs for a really, really long time until the patient unfortunately dies. So we wanted to understand why that's happening. And now I'll take a quick detour from science and tell you all that this was part of my master's research, which was also the first time when I had to move out of my house. And I had to do the most horrific thing ever, which was to cook food for myself. And I know half of you guys know what I'm talking about. The kitchen just takes up so much of my energy that I simply wanted to, find loopholes so I wouldn't have to cook for myself. Zomato still didn't sponsor me, so what's the point? Surprisingly enough, this bacterium over here utilizes a similar strategy to continue chilling in these lungs. Let me expand on that. So this bacterium is very clever, okay? And it realizes that the thick, sticky mucus that it is surviving in has free-flowing amino acids and nutrients, etc., etc. So it thinks to itself, if there is free food in the environment, why bother making any, So it just simply sits back, relaxes, saves up on a lot of energy that it would usually spend on cooking its own food, and simply just takes it up from the environment. Now, this saves this little bit of saved up energy, does it really matter when we talk about disease dynamics in the hospital settings? So we ran a few infection relevant statistics to understand if this was making a difference. And we saw that this bacterium, by saving up that energy from not cooking its own food and by chopping off genes that were initially required in producing this food, save up on energy, which leads to higher maximum growth rates, which also leads to them persisting in these lungs for a really, really long time. And because it has a lot of energy now, it's also stronger to fight against these antibiotics that are given to these patients. So we found out, how these patients survive in these lungs for so long despite antibiotic therapy. And where does this study really take us? So number one, this bad guy over here, it's called Pseudomonas. We understand the evolutionary mechanisms of this bacterium. We're one step closer to understanding it, not just in respiratory, but all the infections, because its metabolic adaptations have not been looked at before. The second thing is that because bronchiectasis has been neglected, so this is one of the very few yet upcoming direct studies for bronchiectasis. And ultimately, I just want to say that research scientists working on respiratory conditions from all over the world are working for the betterment of respiratory health. So don't worry, sit back, relax, and take a deep breath in and out. Thank you.
Can we have the questions?
So it sounds like the best way that you have found to sort of evict this bad tenant is by starving it. What other kinds of potential strategies do you think there might be?
Right. So one thing obviously is metabolic adaptation, but something that's completely different from, instead of starving it. Another thing is that we can use viruses that infect bacteria. So they're called phages, and they infect bacteria. So our lab is, in fact, working on something like this within the same study. So we're utilizing viruses to sort of target these cells, these metabolically adapted cells, and then finding them and killing them. So that's one thing that we're looking at.
Such a fun answer. Just finding them and killing them.
So you were saying that you listen to other people's conversations.
I do.
Yeah, now that I'm listening to your conversation along with the previous TYT one and saying, hey, how about you take that nano particle and that nano material that she had and use that instead? Isn't that easier?
Absolutely. I think that is one thing to go about diseases. I think her research is fantastic. But unfortunately, because bronchiectasis has been neglected so far, I mean, what's happening with bronchiectasis patients is that we're extrapolating results from diseases that we've been assuming are similar to bronchiectasis, and then, applying that. So one of the big reasons why we did this study with bronchiectasis patients and not cystic fibrosis, for example, is because they've been neglected really for a really, really long time. So we need more direct studies on bronchiectasis to understand at what stage would it be beneficial if we were to remove the bacteria in the first place? Because it is playing a lot of dynamics, and this was part of an evolutionary biology study. So a lot of factors come into play. So I wouldn't say that we can directly apply to it. I'm sure that is one potential answer, but as of now, no, there have not been enough direct studies.
I agree.
I'm glad you do.
Yeah. I think you're making sense there. Yes.
Absolutely.
Exactly what I taught you.
So how generalizable are your results? Is question number one.
Right.
How much can you extend it to other pathogens?
Right.
And then question number two is, I'm sure you know that we in India are the largest consumers of end-of-line antibiotics.
Yeah.
And if you to extend that, what it means is that we have a large percentage of people who have developed resistance to several antibiotics, right? And I'm sure we've had some talks about that in these 2 days.
Yeah.
As a policy, have you thought of some of these ideas of what we could implement, or how do we reconfigure our consumption?
Right. So your first question was how generalizable the study is. So if we talk about bacterial infections in general, there are three very important stages that the bacteria has to go through in order to establish an infection. The first one is producing virulence factors. The second one is invasion. And the third one is metabolic adaptation. So it's not just exclusive to Pseudomonas. A lot of studies have been done on this metabolic adaptation of different kinds of bacteria. So I think it is very generalizable in terms of looking at these metabolically adapted bacteria, which we call auxotrophs, as potential biomarkers of understanding how far the disease has come. So another thing that I unfortunately didn't have the time to mention in my talk was when we compared patients with bronchiectasis who had Pseudomonas infections, the patients who had these metabolically adapted bacteria were actually more sick. So it's not just exclusive to Pseudomonas. It's metabolic adaptation is going on in basically every bacterium out there. So, and the second thing which we talk about in terms of antibiotic resistance, unfortunately, India is the consumer of end-of-line antibiotics. So I think when sort of phage therapy comes up, I think that's something that we need to look at because Pseudomonas is also one of the most, it's a highly critical pathogen. So it's part of the top 6 pathogens who have become super antibiotic resistant. So I think we need to move out of antibiotic resistance in general and move to other things such as using phage therapy.
Well done. Zordar taaliyan.
Thank you.